Okay, thank you again for having me here and for all of you for attending this. So I'm going to speak about something fairly different than what I spoke about on Tuesday. So I'm going to talk about work on analyzing and modeling time series gene expression data, and in particular two methods, STEM and DREM, that I'll explain. So to motivate this, a lot of biological processes occur over time, from immune response and development, and often these processes, they're studied with a time series of gene expression experiments where microarray or now RNA-seq are done in consecutive time points. And many methods have been focused on modeling and analyzing gene expression data and constructing networks. And in many cases, they even have used time series data to construct these networks or representations, but they've ignored the sort of temporal nature of the data and just at the end of the day give you a static representation of the situation. So the work I'm going to sort of try to show you is some ways you can think about the time series gene expression data sort of preserving the temporal nature of the data. So the first method I'm going to talk to you about is the short time series expression miner, which is more of a sort of a clustering pattern recognition method, and then the dynamic regulatory events miner, which sort of reconstructs, in effect, dynamic regulatory networks for specific responses. So to sort of the focus why I'm specifically talking about short time series gene expression data was, at least when this work was being conducted, and I think it's still more or less the case, that most time series gene expression experiments are relatively short, 3 to 8 time points. This was a distribution of a major microarray time series, or microarray database showing the distribution of time series experiments in terms of the number of points when this was conducted. And then showing also in general, when I surveyed the gene expression omnibus, about a third or so of all the data sets in there were of a temporal nature. And when I'm saying time series, there can also be other types of temporal or experiments such as dose response. As long as the experiment can be in sort of well-ordered notion, a lot of these same techniques apply. And another sort of issue of, why we're interested in clustering is that genes that have similar expression patterns are often involved in the same biological process or co-regulated. But why am I sort of developing a method specifically for short time series expression data? Why can't we use normal clustering methods? And one is having, so if we use methods that have been specifically designed in the time series literature, they aren't necessarily effective for very few time points, like fitting autoregressive models. And then if we use commonly used clustering methods, such as like a k-means clustering method or hierarchical clustering method, they're not using the temporal ordering of the experiments. And potentially also sort of more importantly, when you're profiling thousands of genes over a very few number of time points, there could be many patterns that you could observe just by potentially random chance, because there's a limited number of sort of possible patterns. So it can be hard to differentiate sort of from what's a real and what's a random pattern. And to illustrate this, you could see sort of 3 clusters here, but it turns out that these were clustering from applying k-means on simulated noise, where all the values were drawn from independent identical distributions. But you could still return clusters that sort of visually look coherent. So we took a new approach to analyzing time series expression data, sort of leveraging the fact that we were focusing specifically on a small number of time points, where it made it possible to define sort of reasonable set of temporal profiles that we would expect to see independent of the data that would be both representative of any profile and such that each profile was distinct from one another. Then we assign genes to the most closely matching profile, and then we can compute the expected number of genes per profile, and this is sort of, I'll explain in detail each step, based on the fact that we had the temporal profiles defined independent of the data, and then we can determine the statistically significant profiles. So to illustrate this, what we were doing was we were defining a set of distinct and representative temporal profiles independent of the data, and this is based on the shape of the profile. So each one of these boxes here corresponds to one different temporal profile, and the line is showing you sort of the shape it represents. And these profiles were defined independently of the data. You don't even need to sort of give a data set to get these profiles. You just sort of specify the number of profiles and the number of time points. And there's also a unit change parameter of the maximum unit change between any two time points. So how do we actually come up with this set of profiles? So we initially can enumerate all possible profiles which change at most 2 units between time points. So this is that complete sort of space of possible profiles. But some of these profiles are fairly similar to each other. So what we actually want is a distinct and representative set. So we have a greedy algorithm which computationally takes one of the, sort of the most extreme profiles and then iteratively selects the profile which is most dissimilar to any profile that we currently have based on a correlation-based measure. So we can continue to iterate and so on until we sort of select these subset of profiles which are designed to capture the space of possible shapes that one could expect to see from a time series experiment for this limited number of time points. As the number of time points grows much larger, this type of approach wouldn't be as effective because it would be hard to get sufficient representative coverage. But because most time series are relatively short, this turns out we can leverage that here. And then what we do is we take the genes that we have and we assign them to the profile that most closely matches their shape. So each of the genes is assigned to the one profile which is most consistent with its shape. So for example, here you see the number in the lower left corner is the number of genes that were assigned to this. And we'll filter flat genes and so non-differentially expressed genes, and we also filtered the flat profile. And I'm showing you data for an immune response to pathogen infection. So we had 150 genes assigned to this specific profile, for example. Now we can leverage the fact that these profiles were independent of the data, and we can ask how many genes would we expect to see assigned to each profile if the ordering of time did not matter. So we're going to permute the time points, do lots of different permutations, or every possible permutation of the time points, and reassign genes to profiles. And then, so this is showing you an example of the time series data under the real ordering of time, and then under one permutation, we would do lots of these permutations. And then under all these permutations, we see that on average there's 28 genes assigned to this profile of 43. So then we can ask, is the number of genes that we saw under the real ordering of time significantly greater than under a random ordering of time? So then it's sort of saying there's something meaningful about having so many genes have that pattern under the real ordering of time. So we're gaining the statistical power by profiling large numbers of genes. So we can do a standard binomial test to associate a p-value with the number of genes that were assigned to profile compared to how many were expected. And I've colored those profiles which were significant, and there's a p-value in the bottom left corner. And then we can sort of do a post-processing step where we can group similar profiles that were significant into the, give them the same color. And then after we do that, we can analyze these profiles for enrichment for various biological categories, such as from the gene ontology annotation. So not only was this profile significant in the number of genes that were assigned, but there's a significant enrichment for genes annotated to negative regulation of cell depth genes. While in this profile, there's a significant enrichment for DNA replication genes, which would make sense in an immune responses experiment. You would, the cell might start shutting down the cell cycle if it was under attack. So now I'm going to give you a demonstration of this method, and it's been implemented in the short time series expression miner, and this software is designed to be sort of very user-friendly, where an experimental biologist could easily go in and start gaining insights to the analysis. So the data file would look like something like this, where you have the first column would be a unique ID, and then optionally you could have a second column with the gene name, or you could just have one unique gene name in your first column. And then you have consecutive columns which have the different time point headers and then the expression values. And I checked this check mark here because I had that first column with the spot identifiers. And then we could view that data file with that button that was input from this interface. Then there's a couple options here. This is either log normalize, or sometimes people would refer to this as log transform. Ideally, you would want this data, when it's applied to this method, to be in sort of log units. So if your data is not already, and everything's sort of relative to time point 0. So normalize would just sort of do a subtraction of everything relative to time point 0 if it's in log space already, while this would do a log ratio versus the first time point, and this would just add a 0. And for all these options, there's these help buttons, which you just click the help button, and then it explains the various options. So it's right there in it. And if you go to the URL that I had in the PowerPoint, there's a very detailed manual explaining all the options. And then if you have multiple replicates, there's also ways to incorporate that, which is explained in the manual. And one of the, I think sort of what makes STEM very useful is that there's a tight integration with the gene ontology, where you would need to specify what you would want to use as your annotation source. And it has built in most of the standard annotations. So this is sucking in the annotations from human EBI. And when you first run it, it'll download the annotations and the ontology. And then if you want to get it updated, you would need to check that box again, and then it'll re-download and overwrite what you currently had. And then there's also an option to include a chromosome viewer, but I think that's sort of a secondary usage. And then the default is to use the STEM clustering method. I had also put in using the same interface if you wanted to just run a standard k-means, but you don't get sort of the unique method here. And then there's sort of two major parameters which define the set of profiles you have. So this is the maximum number of profiles, so the default is 50, and I think generally just using that is fine. And then also the maximum unit change, which sort of controls how much between consecutive time points shape can change. And again, I think 2 works reasonably, and don't necessarily have to tune these, but you could try. And then there's a number of additional options for gene filtering. And you can do gene filtering outside of STEM if you prefer. And then some more advanced annotate options that are described in the manual. And then there's also some options related to the gene annotations, such as if you only wanted to use annotations with certain types of evidence codes or biological processes, or just molecular function or just cellular component. And then also like some categories of where in the Go hierarchy you would want to cut off from, or the number of genes. So I guess I didn't explain this. So the gene ontology, for those that aren't familiar, it's a curation of lots of, I mean, from the literature, there's lots of human curators annotating genes into a well-structured ontology. So then once you've set all your options, you can execute. There's also an option to load previously saved settings. I had already run it. It actually only takes a minute or two to run for most time series, but just to save those minute or two, I'll bring up the output that I had already sort of preloaded. And it's on an interface which is sort of movable, and you can even zoom in. So what you're seeing again here, the colored ones were those that STEM felt were significant. If you do order by significance, you would actually see the p-values associated with it. And if you click on any of the profiles, it'll bring up a plot of showing the individual gene expression values. And then you have two additional buttons here. One is the gene table, which brings up all the genes that were assigned to it, and then their expression values at each time point. And then you have the option to save the table to a file, copy the table to the clipboard, or just copy the gene names, or just save the gene names as well. And if I had checked the chromosome viewer, there would be an option to load these onto a chromosome viewer. And then the other option is you can click the Go table. And then what that'll do is it'll immediately bring up a gene ontology enrichment specifically for those genes that were assigned to this profile. So you can see it's pulling up a lot of cell cycle related categories. And it's telling you some statistics. This is the number of genes annotated to this category of the set that was being considered, the number of genes that were assigned to this specific profile from that category, the number that would be expected based on the size of this, what's the difference or the enrichment, the p-value, a corrected p-value, and the correction can either be done based on a Bonferroni correction or a randomization correction. The default is a randomization, and then the fold enrichment. And then if you click on any of the rows, it brings up the individual genes that are contributing to that specific category's enrichment. Was there? And then for, there's also, for those that are part of profiles with multiple in the same color, that's when you have the cluster gene table, and that's gives you the table of all the genes assigned to those significant profiles of the same color. But it's otherwise the same type of table in Go analysis. And what can be useful is this order by profiles option. So sometimes you might be interested in some specific biological category. So say we were interested in cell cycle, you click there, and then it orders the profiles by the relative enrichment for cell cycle genes. So what it's telling you is that this profile had 24 genes that were part of this cell cycle, and that was the p-value in terms of the gene ontology enrichment. There's another profile that goes down that had 4, and so forth. And then you can click, sort of scroll through other categories and then see the various profiles that are coming up. Yeah. So, I mean, so there's been a couple other methods that have been designed specifically for time series. Some have been designed more for longer time series, and then there have been some which have been focused more on like the differential expression question. And I know like John Story's group also had a method specifically on this. So this isn't addressing like differential expression, though there's actually there is a way to compare, let's see if I have this going, 2 data sets. But I think I'm not remembering offhand like the exact, all the functions that are in that package explicitly, but it's, I mean, my guess is they're doing sort of have some different functions.
And I like the visualization too, so can I use it for case control, like putting the, I don't know, the control like 0 or case, whatever, just translate the case control study into just to.
So there's the k-means. If you're interested in doing k-means clustering, not the sort of STEM-specific method, then if you went to the original menu, then you could run a k-means clustering. One issue is that the first time point might always still need to be 0, though. Yeah, but it wasn't really designed to be a all-purpose gene expression analysis tool. It's designed to sort of optimize for time series analysis. Yeah, so this isn't quite as often used, but it's to let you know it's in here is the comparison option. So what this allows you to do is if you went to this compare button, what it did is it ran a STEM analysis on another data set. And then it's showing you the profiles that were significant for this. And then what it's showing you here is of the profiles in the first experiment, what profiles did they have a significant overlap with in the second experiment in terms of common genes. So it's showing you that there's a lot of common genes in the first experiment that were had the same profile in the second one. And then you can click here and get the list of genes that were common between the two experiments that were both assigned to that same profile. But in some cases, there might be profiles that have a different profile that still have common genes as well. So you might find in this way some specific pathway that's behaving differently between two experiments. So STEM allows you to visualize that. And then there's also some other options, like you can pull up a master table of all the genes and get a list of which profile each gene was assigned to. You can also, there's a number of interface options, like you can display all the genes, or right now I'm only displaying genes that are part of cellular metabolic processes, for example. And then you can also change, sort of play with how the axes are scaled, whether it's uniform or based on the actual time in terms of the visualization. In the actual method, STEM doesn't take into account the duration between time points. It assumes that they've been sampled at a sort of biologically, the uniform rate is sort of equal biological interest between each interval. So I think I'm going to move to DREM unless there's questions about STEM, or you can ask also at the end of the talk too. So, okay, I'll keep moving, talk about DREM. So DREM is the dynamic regulatory events miner. And so what STEM is effectively doing in a lot of clustering methods, what you're getting are oftentimes independent patterns. But there's actually a lot of sort of overlap in the sense that these patterns might have shared the same expression until some time point, and then there was some divergence where a set of genes, some pattern had a higher expression and some genes in a different pattern had a lower one. So we were interested in explicitly modeling this in terms of bifurcation events. And the other thing we were interested in doing is integrating complementary static transcription factor gene interaction data to start gaining mechanistic insights into what transcription factors might be driving the gene expression data. So so transcription factor gene interaction data is represented here as a binary 1 or 0, that this transcription factor regulates this gene. And this could be derived in a number of ways. It could be derived from ChIP-seq experiments or ChIP-chip experiments. So these are binding experiments where you map where transcription factors are binding, and you can ask if they bind within so many kb of a gene promoter, for example. They can be predicted based on regulatory sequence motifs. If you know a pattern in the DNA sequence a transcription factor recognizes, you can computationally predict it regulates it. For some model organisms like E. coli, there's a fairly robust curated network of transcription factor and gene interactions. So we're taking effectively the same genes where we have the time series data and then the static transcription factor gene interaction data. And in effect, the method is going to project, even though this isn't temporal information, sort of elevate it into dynamic contexts.
Do you distinguish between upregulation and downregulation?
So yeah, the method, like in the E. coli, when we do those, we can have sort of three, a 1, a 0, and a -1, where it's known that this is an activator or repressor or not. When you're doing predictions from like regulatory motifs or ChIP-seq experiment, it's hard to know if that binding is an activation or repression association. And if it's the case that it's sort of always an activator or always a repressor, it's not much really lost by just putting 1, 0, where it's important is if it's sort of half the time working as an activator and half the time working as a repressor in the same cell type. That's where it can be very useful to know the direction in terms of the modeling here. I mean, in general, it would be useful to know, but where it would sort of affect the actual model that would result. Okay, so this is an illustration of gene expression data for heat shock response in yeast of the raw data, and I'm going to illustrate sort of the modeling we're trying to do. So what we're effectively trying to do is build a map on top of this expression data, highlighting the major regulatory paths and bifurcation events, and then annotating them with potentially transcription factors responsible for them. So when I have this arrow here, what it's showing you is the set of genes in orange were assigned to this lower path, while this set of genes in gray were assigned to this higher path. And these set of, DREM is making this prediction that these set of transcription factors are potentially driving this set of genes higher, while these set of transcription factors might be driving it lower. And then we can follow this lower path here and see another bifurcation event, another one here, and so forth. And in some cases, we can also model trifurcation events. And now I'm going to blow up this specific example, and you can see here that there's a sort of nice pattern in that these set of genes have effectively the same distribution until somewhere around 60 minutes, and then there's this clear separation over the last two experiments that these genes had a higher expression pattern than these set of genes. And what I'm showing you is that the genes here are those genes that are regulated by MPP1 based on the static input. So these genes I'm showing you are just the genes that were regulated by MPP1 based on the transcription factor gene interaction input data. And you can see most of them are on this higher path. And so what we can do is sort of make this prediction that these transcription factors, and there were several other, were regulating these higher genes. And if you're familiar with yeast transcription factors, you would recognize these as the cell cycle transcription factors. And in collaboration with Idamar Simon's lab, they went and did so this was in heat shock experiments. They did a bud counting experiment to allow you to count sort of whether you had no buds, small buds, or big buds to get a sort of quantification of where things are in terms of the cell cycle. And it turned out that there was a spike in the no buds at the same time. So this was reflective of sort of a resynchronization of the cell cycle after sort of recovering from the shock of the stress. So this is an example of how you can sort of model this and then also get experimental confirmation of what you're observing here. So to give some more details about how sort of the underlying model behind DREM, I'm going to illustrate this with a toy example. So say we just had these three paths which had the same expression, then diverged, and then diverged again. And we're going to sort of model them as having a Gaussian distribution of expression along some path. And then if we didn't have any additional information about the transcription factors regulating the genes, we would say the transition probability, if we're assuming an equal number of genes on each path, 2/3 probability to go up here, 1/3 to go here, 1/2 and 1/2 would be the most likely probability. But DREM is a little more sophisticated in that it also models the transitions to take into account what we know about the set of regulating transcription factors for a gene. So if we knew, for example, that most of the genes that were on this path were regulated by transcription factor A, so genes regulated by transcription factor A tended to go this direction and then this direction, then if we had a gene which was regulated by transcription factor A, we would want to put a higher probability that it would go up on this path and then a greater probability to go on that path. And similarly, if we knew genes regulated by transcription factor B tended to go on this path, we would put a higher transition probability to go down this path. And we can more generally convert the set of transcription factors that bind or associated with regulating a gene to a gene-specific transition probability with a logistic regression classifier. So it takes this vector of the transcription factors that binds to genes and then converts it into a probability that it would want to go on the up path or the down path just based on that information. And then we more generally model it with a sort of a very constrained instance of an input-output hidden Markov model, which enforces this tree structure and assigns each gene to its most likely path based on both its expression distribution and this TF gene interaction transition probabilities. So that's the underlying model. There's a lot of possible tree structures. So what the software, the method does is it does a search over possible tree structures and penalizes it in terms of also complexity on the model structure. And this is an expression for sort of the scoring in terms of the likelihood of the data. It's somewhat similar to the HMM work I had presented before, but it's a little more with this extension that the transitions can depend on the transcription factors themselves that bind. So this is another example in yeast of the output. So this is looking at amino acid starvation response in yeast. And what you can see is a tree structure labeled with, again, predictions of different transcription factors. And it finds some of the major amino acid starvation regulators here, and then also some ribosomal factors down here. But then it was also making a novel prediction. So INO4 was not previously known to be involved in amino acid starvation response. How we have the transcription factor gene interaction for this was largely based on its binding of genes in standard growth conditions. So in this case, we have condition-specific time series expression data, and then binding data which doesn't perfectly match the condition, but the hypothesis is if there's enough overlap of the target genes that it can still be informative. So what we did was to sort of validate that this transcription factor was potentially having a role in amino acid starvation response, in collaboration with Idemar Simon's lab, they did a new chip-chip experiment for INO4, specifically in amino acid starvation response. And what this is showing you is that there's more intense binding in amino acid starvation response for genes on this specific path that was predicted to be controlled by INO4 than in sort of standard growth conditions. So this provided experimental validation of a computational prediction made by the method. I had also applied this in context of the ModEncode Consortium's work looking at the fly development, and also a model in collaboration with Zoltan Oltvai's group looking at aerobic, anaerobic shift in E. coli. And then there's been many other applications of DREM published by others. And I'm going to now give you a demo of DREM. So the input to DREM, it's actually pretty much the same format in terms of the time series as STEM. So you have in this case, the gene IDs and then different columns for the expression values. And then there's that same spot variable if you had if you had a spot identifier as well. In this data set, we don't. Then there's a number of built-in transcription factor gene interaction sets based on a variety of different sort of methodologies. Like for E. coli, it's there's both a curated set and also a set based on extended with computational predictions. For fly, there's one, this was based on the mod encode, built a network, integrating a lot of different data. For human, there's sets based on some encode chip-seq data, and also predicted binding based on motif scanning. In yeast, there's data sets of transcription factor gene interactions based on the work of from Rick Young's lab, primarily from the chip data. And oftentimes you might want to build your own transcription factor gene interactions based on what's most relevant to your data. For example, if you had sort of chip-seq data for transcription factors specifically in the cell type you're interested in, you might want to use that. And the format of this file, this is showing you the view of it, but it's a fairly straightforward format where you would have the rows would be the genes, the columns would be the transcription factors, and then you have a 1 if it's predicted to regulated, 0 not, and if you wanted to differentiate activation from repression, you would have 1, 0, or -1. And then you can also do a user-provided transcription factor gene interaction data. Then the expression data is as before, and you can also view that straight from DREM to sort of, if you want to check what it actually has. There's a similar thing with, if you had multiple time series for the same experiment, you can load that in. And then again, there's options for how to transform the data. So ideally, it should be after the transformation, it should be in log ratio relative to the first time point. It works like STEM in that it's also integrated with the gene ontology, so you would want to specify your gene annotation source that's relevant to the system you're working with. So here we're working with yeast. And then you can, again, re-download the annotations, or you would be required the first time. And then there's a number of options as well. Some of them are similar to STEM, but then there's a number that are specific to DREM in terms of how it does the model search. And these are described in the manual. But in most cases, you wouldn't necessarily have to modify those. And then also, I forgot to mention here is, in this case, I'm going to load a saved model. DREM, depending on the complexity of the model, can take longer to learn the model, so oftentimes you'll want to save that and then re-specify it here in order to not have to reload the model again. Okay, and I had already opened this. So in general, you would hit execute, but to save us a little time, I've already opened the model. So this is actually the same model as I was showing you before. This is looking at the heat shock experiment. So this is what sort of a visualization of the data, of the time series data. And then what DREM does is it puts on top of this effectively map of the response. And then you can click on the bifurcations to see sort of visualize the path, the split. You can click on individual edges to see a visualization of just all the genes along that specific path. And then for example, if we're interested in some specific split, so I was highlighting before the split here, and you could hide the nodes if you want to visualize it again. We can then click on the set of transcription factor labels or the edge. This would be a right click to bring up a new window. Left click selects the set of genes. And then it brings up information about the transcription factors associated with this path, in the sense that what it's saying here is in total there was 88 MBP1 target genes, and then 42 were sort of going into the path, sort of on the parent node, and then, or into the split, and then on this specific edge, this top edge, 35 of the 42 MBP1 genes were on that path. And then it can compute the enrichment both overall and then specifically conditioned on the split. So there's sort of a difference in interpretation. The overall is enrichment when you sort of, the cumulative enrichment considering all the events that it took to get to that subset of genes, while the split is a very localized enrichment saying of the genes that had exactly that sort of path assignment until this time point, what was the enrichment? So expected is how many overall we would expect on this path. Expected split is how many we would expect on the up path, conditioned on there being 42 from the parent. And then it gives you a score. These aren't true p-values in the sense that the model was learned using transcription factor gene interaction data to learn a more coherent model. There is an option in DREM to learn the tree structure independently of the transcription factor gene interaction data. In that sense, then these would become true independent p-values, but then you might miss some interesting biology because the transcription factor and gene interactions can help you learn a more coherent model. And then all these columns are sortable, so you can sort by different factors. And then again, you can copy or save these tables. So that was a split table. If you're interested in a specific path, you can do a similar thing and bring up a report on just the genes assigned to some path and see the transcription factors sort of associated with genes on that path. And you can also, when I click on a path, I can click the gene table button and it'll give me the list of genes on that path and then their expression values, and then also a report on the which transcription factors were predicted to regulate those genes. And then you can also click the Go Table button and it'll give you run a gene ontology enrichment for the set of genes that are being displayed. And another useful option is, let's do select by TFs. So you can, if you click the select by TFs button, then you can, for example, pick what transcription factor you're interested in, and we're going to say, for example, if we want to know SY4 bound, and then we do apply selection constraints, what it's showing you now is specifically the set of genes that in our input, which were SY4 genes. And then you can even mouse over the specific genes and get their names as well. Let me do. And then, let's see, select by. Right. And you can also select subsets of genes assigned to specific Go categories as well. Yeah.
I realized that at each breakpoint,
Yeah.
you're using the information from the static definition of the transcription factor sensitivities. That's correct, yes.
At each breakpoint, yeah, it's the same. Right.
And I wonder if there's a way to do a kind of internal consistency check, because some of those genes must be the transcription factors. And so then you could ask whether the time course of the transcription factor was actually consistent with the identification you made at a branch point. You see what I'm saying?
Right, yeah. So we have done in some work some analysis on the sort of comparing the expression of the transcription factors to their predictions. I mean, in some cases, the transcription factors are more post-translationally regulated, so that there might be some like phosphorylation change or something where you wouldn't at the expression level necessarily see the change. And I think in some analysis we were finding that the transcription factors, which were like the master regulators that were like first to respond to a lot of genes, tended to already be, might already be expressed. And then some of the later ones might be more fine-tuned. But it would probably depend on exactly the condition you're looking at. So that's most of DREM. Let's see if, yeah.
So for each time point, for each gene, you have only one measurement.
For each time point, for the, in
Each
the,
time point for each gene, you have one measurement.
right, in terms of the gene expression data.
Yes.
Right.
So and what happens if you have more than?
How do you deal with replicates?
Replicates, yeah.
Right, so replicates are just effectively, let's see, so you can add replicate data here, and then it just effectively averages multiple readouts, except it can do an additional thing where it can check if you had sort of a longitudinal study where you're following the same gene across multiple time points. It can filter genes which have an inconsistent profile across time points.
I'm sorry, I don't understand. So how you deal with replicates? So because usually when you do, in the web of something, we have replicates at least 3, 4.
Right. I mean, so it'll effectively average the profiles to just work. Yeah, but it has an option to filter genes that are just inconsistent across time profiles. So if it's basically if a gene is up in one experiment and down in another, you could represent the average as just the sort of middle profile, but then it might be better just to filter that gene.
So follow up on this one. Can I use this software for aging study? So what I mean aging study, when you have 5 aging, age cohort, like from 40 to 60, 61, just 3 of this. And if I'm averaging for each gene, for each samples, can I use this one? Study aging?
Yeah, I mean, as long as you have temporal ordering of your time points, it should be.
I'm wondering what the time series it means, because we are talking about minutes and seconds, and now we are talking about decades. So it doesn't mean anything in terms of your design.
I mean, in terms of the computational method, it would apply just as well. I mean, the biological interpretation of the results in any specific application will likely depend on the application, how appropriate the model is for the specific data. So probably the best way is to try it out. Yeah, and then I was, just to show you again, with the Go enrichments, again, you can project on, for example, this is looking at ribosome and showing you the enrichment for, showing you just the ribosome genes and then enrichment on what pathways they're assigned to. And again, there's various options to play with the interface. A lot of times things can get crowded, so you can zoom and pan and things like that as well. That is with the right mouse, and then you move the mouse up and down. Yeah, and then move the mouse sort of left and right. And yeah, the left mouse, and you hold that down, and then you move left and right, it pans it. And then if, yeah, to save the model, you would go click Save Model, and you can also save your parameter settings as well. And there's a command line option to open DREM with a saved set of command files. Saved set of options. And we have a few more minutes left. So another thing you can do is you can ask, given I had some combination of transcription factors, so say I was bound by SY4 and MBP1, it can sort of predict, according to the model, which path I would most likely be on. So it's saying here, probability 0.92, I'd be here, 0.81 here, 0.6 go on here, and 0.45 here, and 0.32 be over here. So if you were interested in, say I had a gene that would be regulated by these transcription factors, what path would probability I would be on each node at each time point, you can get that information. And then you can also, for those transcription factor labels, you can make the threshold more lenient or more stringent to get a longer or shorter list. And right now the significance is based on conditional on the split, but you can also do it overall where it's taking into account sort of the power from multiple splits as well. And all these options should be described in the manual. And then with DREM, there's actually been, Bar-Joseph's group has published a number of extensions to DREM, for example, dealing with microRNA regulation and integrating some other network information that I haven't specifically been involved in, but there's publications and sometimes different versions of DREM from the main one that implement some of these extensions. But if you go to this website, they'll link to most of those. So to summarize, I presented a method to identify significant patterns when clustering short time series gene expression data, and a method to globally model gene regulation dynamics that integrates time series gene expression data with static transcription factor gene interaction information. And all this work was done while I was, or almost all of it, while I was a PhD student in Ziv Bar-Joseph's group at Carnegie Mellon. And then these were some of the experimental collaborators who we worked with on some of the initial applications of DREM. And DREM has been continued to be developed by a number of other people in Ziv's group after I left. So thank you for your attention. So questions about either STEM or DREM?
So remember you have this button for gene ontology enrichment analysis. And so do you recommend a certain gene identifier to make things easy when you input the data?
Yeah, all right, so it'll depend which organism you're working with. And yeah, so at one point with the gene ontology, there was this cross-reference file where it would sort of give a file which would cross-reference other ones. But I don't remember all the ones that if you don't have that file anymore, what is supported by the ontology. But I think like using the human gene HENC symbols is usually does pretty well. But you can check what the gene ontology annotations, what they include. And there's an option in STEM and DREM to provide your own cross-reference file where you can give it one column the identifiers you're using and the other column what identifier it maps to in another system, and then in doing the ontology lookup, it'll do that mapping for you.
So if we do a gene ontology analysis, then we usually use the Ernst gene ID because the mapping between many mRNA arrays is very well. But if you map, for instance, a gene symbol to Ernst gene ID, then you may get several Ernst gene IDs, which might interfere with other analysis steps. So we do as a standard the Ernst gene ID, and only when we give the results to the users, we map them to the symbol so that they can read them.
Mm-hmm.
But this is only my experience.
Okay, thank you.
What's your favorite application of this tool? What biological result would you cite as a really cool one?
In terms of, I mean, I guess for DREM, I mean, got most into the yeast stress response. I mean, that was the one where I was sort of applying that most, and then had experimental collaborators go and validate it. But then there's been, I mean, other peoples who I had no idea they were even sort of using DREM until I see their work in some nice paper, and that's gratifying as well. But it's harder for me to sort of always appreciate exactly that system.
It's an enormous amount of work. It's really, you must have spent an enormous amount of time on it.
Yeah.
Don't make him feel bad.
Yeah. Well, thank you.